So, 1931, Arthur Fox, Wilmington, Delaware, synthesized PTC, short for phenylthiothiocarbonide. Okay, and here's the molecule. Who cares? Not a test or quiz on this, but here is the shape of the molecule. Okay, now some researchers reported a bitter taste when entering his laboratory because of the chemical produced as a dust. Others, including himself, found it odd that they thought they were smelling and tasting. Sometimes you smell, you taste, it's combined. Okay, experienced no sensation. It's like, why do I not get that bitter sensation you guys are? So he hypothesized that the taste was due to the PDC particles suspended in the air, and that some people were able to taste the chemical, where some weren't. Okay, interesting. All right, so I'm just giving you. So how do we taste? Just a little review. The tongue is covered with papilla, and these little bumps have these little taste buds. Inside these little taste buds, okay, eventually if you get a little smaller, okay, we have these gustatory cells, and these gustatory cells right at the tips, okay, have these little receptors. And these receptors are for the sweet, bitter, salty tastes. And if there is a receptor that connects to the end of these little fibers, it'll cause a signal to go to our brain to give us that response. Okay, if we make this even bigger, not that, although we'll get there. Okay, if we make this a little bigger, okay, at the end of these increased surface area, we have these different types of taste buds. And some of them are for a bitter taste. So to elicit a bitter taste, we now know that the chemical that we're tasting has to fit, here we go with the shapes again, into an actual physical receptor. So if my receptor has this shape, physically, okay, the chemical that's going to elicit that bitter taste has to have something similar that will fit in that shape. And if it fits in that shape, okay, by them combining, those amino acids will actually, these amino acids, they bind there, will attract these amino acids to change their shape again. And that's called a transduction pathway signal we're going to learn. But it's going to start a chain reaction that eventually will fire a nerve to our brain to say, hey, that's bitter. But anything that's close to that shape will elicit that response too. So we have a bunch of bitter taste bugs among the salty, sweet, and so forth. And again, to give you a little, this is a way to look at this. Okay, so this is, again, the transduction signal. How does the signal get to the nerve? Who cares about this right now? But there's a receptor, a physical receptor, who's made by a what?
Mutation.
Well, you're probably definitely true, but it's a protein that has an exact shape. And that shape is based upon the amino acids and what kind of what? Shape this makes when it folds. So, taste buds, you will taste bitter if you've got a receptor on your tongue that has the right shape. If you have a mutation and you have the recessive, we're going to call you the non-taster. If you're the taster for the bitterness, you will have the genes that code for the right amino acid sequence that makes the protein in a certain shape for you to accept that bitter chemical to elicit that response. If there's a mutation in my DNA that makes the wrong amino acid sequence, which will make a different protein shape, you can't pick up the bitter taste. And that's the recessive allele called a non-taster. Okay? So, again, I can't teach you this stuff without showing you how it all works together. So, soon after its discovery, geneticists determined that there is an inherent component that influences how we taste. Now, today we know the ability to taste is conveyed by a single gene. They call it TAS2R38. It's a single grouping on a certain DNA and a certain chromosome that makes what? A certain amino acid group into a certain protein that gives me the bitter receptor, the acceptor that accepts that chemical. And what we do know is that if you can accept and taste PTC, you can actually you are able to get the bitter response from other chemicals that have very similar shapes. And there's an evolutionary type of thinking that goes on here. All right, now there's two forms. There's the one that's working, you're the taster, you'll taste the bitter. The one that's not working, you'll be the what? Homozygous recessive. Now, here's what's really interesting. The heterozygotes, it's hard to discern who's a heterozygote or not, but you will taste it, but not as strong. So we're just going to measure tasters versus non-tasters, but the heterozygotes can go either way. Because you may have some being a heterozygote means you're going to have some receptors that are right, but some receptors that are what? No. Right. So depending on how they're spread apart, depends on how well you taste it. Okay? But, in any case, let's continue on. Christmas in July. So moving on. It's been suggested that the ability to taste natural chemicals similar to PTC help human ants stay away from toxic things. A lot of toxins and poisons taste bitter. So if you can taste it bitter, maybe that was a selective advantage to say, hey, that's poisonous. But we have some really good foods that are good for us, including us, the Brassica raptus, right? Yes, plants? That have bitter compounds in them, like cabbage is bitter. So if you taste that bitter, maybe you stay away from toxins, but you'd also stay away from foods that are healthy. And that's why they call it the heterozygote advantage. We'll talk more about it. Being a heterozygote, you can taste the bitterness to say, hey, maybe it's poisonous, but then you can also accept foods that are what? Good for us and bitter. And we'll talk about the heterozygote advantage. Okay. Moving forward. Let's see who is tasters and who is not. Okay? So what you have in front of you is you have two kind of vials there, and these have a lot of tasting papers. Okay? Now, what you do is very simply, we're going to start with the one that doesn't have the yellow paper on it. Okay, now, there's a bunch of papers. You take the forceps. Now, with the forceps, you're going to pull out one. There's one for two people. Or, I'm sorry, you're going to share one of these vials for two people. You'll do your own. Now, when you pull one of these out, try to grab one. I know it's kind of hard. Do your best. If you grab two and two come out fine. Grab one, like I did. Okay? You're not going to taste on the what?
Taste on the metal side.
Metal side. So you're just going to taste part of the paper. Right? Just the middle part. Okay? You have your own there. Okay? So just put this part in your mouth. Okay? This part you just put back into the plastic. Okay? And so even if you use someone else's what? Forceps, you're always working from the end that you're not putting in your mouth. Right? Only half of it's going in your mouth to taste. Okay? So everyone try to taste from the non-yellow. Yeah, when you're done, you put it in there. We're doing the non-yellow first.
Yeah, that just tastes like.
What's that?
Like
you put it back into the plastic tray. So who's got any taste from the first one with this doesn't have the yellow marker on it?
It tastes like paper.
Did everyone taste it? Everyone taste it? Make sure everyone tasted first. Okay, the one that I made you taste is the control.
Wow.
Just to make sure that there's no liars out there, so you guys are good, because there's always a tryhard out there. Yeah, man, I'm a taster! Like, loser! Okay, but you guys did all right. You guys did all right. So yeah, I gave you the control first. Okay, so no losers out there. Okay, now, the yellow one, the one with the yellow marker, that has PTC on it. Okay, so if you don't taste it, you should taste the paper again. But if you taste a slightly bitter flavor, you're a taster. If you don't, you're a non-taster.
Bro, it tastes sweet.
All right, I guess Gavin's a taster. He does not like cabbage. You like cabbage? See? There's something to that. But maybe you're malnourished because you don't eat cabbage. If you don't taste any bitterness out of the yellow vial, then you are a non-taster. If you did what Gavin did, go, ugh, ugh. Okay? So for yourself, you're going to check taster or not. I'm going to test myself too because I don't even know.
You don't remember?
I taste it. I'm kidding.
It wasn't that bad. I didn't expect it to be a lot worse.
I'm a non-taster.
Yeah, 3 in the mouth. He goes, only take.
Oh, wait a minute. That took a while. Yeah.
It took a while with me.
Yeah.
It was really slight. Well, I
Yeah,
left mine in my mouth for like 2 seconds.
I might be a heterozygote because initially I didn't taste anything,
Yeah.
but now I do taste the bitterness.
Wait,
I'm definitely a taster.
I only put mine in for like 2 seconds.
Well, then go back and get another one.
Yeah.
Sorry, Emily.
Big double, make.
That wasn't, but for you it was really bad, right, Gavin?
Yeah.
And it was only one?
It was bad for me.
There is, PTC is not a natural food, but what I will say is this. There is a correlation between those who pick up the PTC bitterness and those who pick up other natural bitterness tastes. So the PTC has a shape that's similar to other, even though PTC is itself not, right, exactly. So PTC has the same shape that's similar to other things that are natural bitterness things. So people that are bitter for PTC will find cabbage bitter, will find poisons bitter. Again, you can see it's a good selective
Wait.
pressure.
So like you could, in theory, like drink poison and not taste it if you don't taste it.
Correct. But listen, if you're in the wild, not where there's what medicine, you can see it's a selective advantage. Oh, it's bitter. You spit that out. Okay, so there's an advantage. But on the flip side, if you're not eating certain foods that are healthy for you because they are bitter, that's a negative.
It tastes kind of moist.
Okay. So,
Show me this.
yeah, that was really good to have on camera. Thank you. Okay, thanks a lot. Okay. All right, and I know this is biology and all, but okay. All right, so myself, I'm definitely a taster. Okay, so let's do the phenotypes. Okay, we're just going to make this very simple. The tasters are either going to be big T or big T, little t. Right? You don't know if someone's a heterozygote. How about I put that on the big screen? Oh, we almost done like 11 minutes. That stinks. All right, I get it done. Okay, so now, non-tasters, and this is important, people. Focus here. This is the jumping off period. This, to be a non-taster, you mean you had 2 alleles, 2 chromosomes, one from your mom and dad that are both broken and recessive, so you must be little t, little t. That's the biggest thing. That's when we do Hardy-Weinberg calculations, party people, that's the jumping off period. Because that's the only what? Phenotype that only has one what? Genotype. That's going to help you. Okay, continue on. Okay, class totals. Hands up if you're tasters. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So we have 14. Non-tasters. 1, 2, 3, 4, 5, 6. Good. We should have tuition. That's good enough. Okay. Frequency. Now this is the frequency is a kind of like a percentage without a percentage. So we have 20, right? So out of 20, there's 14. Do that math. We'll do that together. Put that on the later of calcs. 14 divided by 20. I guess I could have done that too, yeah. So that's 70%, but we don't put 70%, we put 0.7 to make it like a probability. 0.7. 0, because, all right? Because what if it was 1? What if this came out to be 1? Wouldn't this be 20 out of 20? It's kind of like a probability. So keep it like that. Frequencies, okay, are like the percentage without timesing by 100. Use it like based on probabilities. Of course, the other one's easy, right? Because it's out of what? All possibilities. This is at 0.3. Now, if you want to do percentages, of course, it's 70%. This is 30%. Generally speaking, just for fun, they say 70% of target populations are tasters. So it came out perfectly in our little sample. Which means what? Well, we must be a good random sample. Okay. Now, of course, assuming our class is a random sample of the entire U.S., and our population in our society is large that you guys belong to, there's no random mating, there's no mutation, there's no gene flow, and there's no natural selection. Whatever we get as a sample reflects the entire population because the alleles will stay constant. So let's go on to the next page. This is where I need to really focus down here. Now, so the jumping off point is where? The heterozygotes here. Now, Hardy-Weinberg is a formula used for other alleles, but we're going to use just one, the taster allele. And this is important. Because Hardy-Weinberg looks at a single allele, and it could be different from a taster allele, whatever we're going to look at, it could be the gene for hemophiliacs, correct? We're going to use not big T, little t in our calculations. We're going to change it. We're going to do this. We're going to say Q, okay, is P is the dominant, and Q is the recessive. We just do this because we're going to what? Use different alleles all the time. But we know better. The dominant in tasters is big T. The recessive is the little t. Okay. I still got that bitter taste in my mouth. Okay. Now let's go rewrite this. Now that I know that the dominant is P, so this would be what? PP, right? This would be PQ. This would be what? QQ. It's going to be saying something. QQ. I don't know. The dominant allele is what? P. Recessive is Q. Now, little p helps. Okay, good. Now, let's go back to some rules here. And this is how I'm going to derive this. I could just give it to you, but I want to derive this. So please focus. I know you've been in school all day. Isn't the probability of a zygote's going to be what? Haploid number. We're going to forget about all the other chromosomes and all the other alleles. Isn't the chance of that zygote, either get the mother or father's, isn't the chance to be P and Q? I don't know what the percentages are. Right now it's 0.7 and 0.3, right? But since there's only P and Q, what are the chances of a zygote having a P or a Q? What are the chance of rolling a heads or a tail? Right, so P plus Q equals 1. That's how you figured out the other one. So this is important. You with me? Good. Now, this is where the leap comes in. Stay with me here. If I have the male gamete, what is the probability of having a P or Q? Total 1 here, but there's a chance, right? This frequency, remember, this is important. Little p is the frequency of just the P allele, which we now know is the dominant, right? The big T. This is the frequency, remember this was what? We don't know this for sure, but this is the frequency of just the recessive allele. What did we find? Really important. We found, on the other page, sorry, we found, Christmas in July, the frequency of the individuals. We didn't find the actual T, right? This is what? Let's rewrite it. This is really what? QQ. This is what? PQ. Or PP. So this is individuals. Okay, cool. Now let's go back to the page. Stay with me. Now, we want to find a formula. Well, listen. This is a separate event to make this gamete, isn't it? And this is a separate event. This is the probability of having a P or a Q. Equals 1, right? So I multiply them together because remember we learned about the multiplication rule? Separate events to make gametes. Gametes come together, make what? A zygote. So let's do the what? I learned this as FOIL. First, P times P is what? Okay, P times P is P squared. How about outer? P times Q? PQ. PQ, you are the best in your area. Okay, then we have inner. Q and P, PQ again. PQ. And then we have last. QQ squared. Q squared. Now put this together, party people. PP squared plus what? 2PQ plus Q squared. Now party people, that equals 1. Now these are individuals. I'm sorry, PP squared. It'd just be P squared. I'm being too many P's here. My bad. I feel bad and I'm over it. Okay. Now, who do you think is P squared? Homozygous dominant. Who do you think is Q squared? Recessive. What's my jumping off point? I don't know these individuals. I don't know who's homozygous dominant. I don't know who's a heterozygote, right? Aren't these blending together? Who do I know? Don't I know Q squared? What was the value for Q squared, party people?
0.3.
What's that?
0.3.
0.3. Now that was for what? That was for the individuals. I want to go for Q. I want the recessive allele. So if I know Q squared, which is 0.3, Q is equal to the square root of Q squared. So let's go find the square root of 0.3. And I get 0.547. And that equals what?
0.6.
Now you can't round here. Equals Q, right? What is Q? The frequency of that allele in the population. Can we get the frequency of the P? How do we do that? P plus Q equals 1. Subtract by 1. And I get the P to be 0.452277. This is really what? Don't leave. This is what? Little t frequency, big T. Can anyone guess? How do I get the P squared individuals? Do what with that? How do I get my homozygous individuals? Right, plug it in. Please do that. I'm going to post a lecture to finish this off. So, okay, I want to finish this off together, and you should get this, okay? Now, please write this in, because understanding these little differences will really help you. This P and this little Q represent the frequency of all of the population's dominant alleles and all the population's recessive. It's really the probability, right, when you think about it. Okay, and I want you to understand that little p really represents, hey, how many T's are out there? And little q represents, in the entire population, how many little t's are there, right? We're not counting individuals. We talked about hemophiliacs who are homozygous recessive for that 2 broken alleles for the blood clotting factor. Okay, those individuals. We want the individual, okay, frequency. And I get a frequency value, not a percentage, it's out of 1. Okay, so we found, okay, the individuals that are what? Homozygous recessive to be 0.3. 3 out of 10 was basically what we found. Okay, but that's not what this is. This is Q. All right, so it's very important. Okay, you understand that difference. So the frequency of the P's in society and the frequency of the Q's in society, okay, have to equal 1 for the development of the gamete, the haploid cell. What? With haploids are either going to get a P or a Q. So these are all the possibilities. These guys are mutually exclusive to each other. So they both have to equal 1. Okay? Now, when we mate this with another gamete who happened independently of making this one from a female gamete, they have the same scenario, that the total what? Combinations of P's and Q's will have to equal 1. Because making the female gamete, you're either going to get a P or a Q in that gamete. It's one or the other, so the possibility is still 1. And because we're now going to mate the male and the female, okay, we add all the possibilities that this single chromosome could have for this allele, and this has the same scenario because there's only P's and Q's. And because the making of this gamete is independent of this one, we multiply, right? Because the meiosis that occurred for this male in spermogenesis is different from oogenesis, the egg that was made. And so we multiply these. And so when we do that, I know some people do it differently. I used FOIL, okay, when I was growing up. I don't know what the New York State math is now. But I'm going to do first, right? Binomial, P times P, so the first. So that's going to give me P squared. Okay, then I do outer, P times Q. These are my outer ones. So that gives me plus P and Q. And then I do my inner. Again, P times Q is again PQ. And then I do last. Q times Q, and that's Q squared. And if I simplify, I get P squared plus 2PQ plus Q squared. Now this equation equals 1, and this is so important. Okay, so I'm going to change colors. Okay, this is for individuals. We can use this now. This is the Hardy-Weinberg equation for individuals in a population if there's no what? Small population, no selective mating, if there's no mutation, okay, no influx of gene flow, and no selective pressure. Okay, this is how you figure it out. So we can figure out exactly how many homozygous, because right now it's P and P. Don't forget, the little p represents the dominant allele. So let's put that back in there. So we don't want to lose sight to what this all means. So yeah, we'll stay with red. So this guy really is TT right here. That's what P squared is. Remember, the P represents the dominant allele. Who is 2PQ? That means you just have 2 times, okay, 2 times what? Big T, little t. And Q squared, 2 Q's, remember the Q's represent the what? The recessive allele is the TT individuals. Don't forget the 2 is there to times it by 2 to make it even, but party people, okay, there are your 3 possible what? Genotypes, okay? Either the homozygous recessive, the heterozygote, or the homozygous dominant. There it is, okay? And so, again, I don't care, Mr. Kratz, what's the big deal? Well, I can see if, in fact, we're in Hardy-Weinberg equilibrium, if our class data reflects the data that we have in society. We kind of mentioned that 30% of society is non-tasters, which means 0.3 is my frequency. Now, from my leaping, which one of these do I definitely know? And this is important. My leaping off or my point that is we always take off from is from homozygous recessive. Now, if you look closely of my math here, that gives us the equation to figure out the frequency of these types of genotypes, okay, using this Hardy-Weinberg formula. Notice there's what? A 1 to 2 to 1 ratio of those types of individuals. Notice if we would do a simple Punnett square of a paternal and maternal, okay, you'd get the what? 1 to 2 to 1. And notice this is the leaping off, the jumping off point. And the reason I say that is because if I know this guy right here, we know Q squared. We found Q squared. What was Q squared? Go back to the original. Go with me. Go back. Flip this page over. Okay, let's go back to this page. Please flip that page. This, my friends, was Q squared. I should write with it something I can write. Here we go. This is Q squared, the non-tasters. So we have, okay, I'm going to change this up. Yeah. We have the Q squared frequency right here. That's what we can get by doing this activity. We found the frequency of my Q squared or my homozygous recessive individuals. We have that. And all I have to do is solve for the individual P and Q and fit them into the formula to figure out my other genotypes. I cannot figure these guys out because the tasters were either or both of these. I can't discern between these. So our jumping off point is the homozygous recessive. They're the one that are expressing a phenotype, okay, that's so different from the others, and they only have one genotype. I can't say that enough. You're going to keep your life so much easier if you understand that this is your Q squared, your homozygous recessive. That's going to make your life easy. So now that I know that this is Q squared, and I know there's a formula for the rest, okay, I can do this. All right, now, let's go back to that page. Turn back. Let's go after what P and Q is. All right? I want the individual P and Q. And again, These would be the frequency of the individuals in society. Notice they have to equal 1, right? These are going to, all these numbers, all these probabilities to be these individuals have to equal 1. These are all the what? 3 probabilities, or the 3 types of individuals, okay? So if I had a 3-sided die, which is kind of weird to be a triangle, I guess, I'm not sure. But the point is, if I rolled it, I either get a 1, 2, or 3. Okay, so it has to equal 1, the addition rule. So we're going to go after the individual allele frequencies first, to go back and solve for what the frequencies of my homozygous dominant or my heterozygotes, okay, I need to solve for my P and Q. And the jumping off point, I know I'm being redundant and tall, is my Q squared. So what I'm going to do is, I'm going to go find Q. And like I said down here, to get Q, I take my Q squared, and again, I know that my what? I'll say it again. The TT individuals, the non-tasters, okay, from the first page is 0.3. But that is really what? Q squared, right? Q times Q. All right? So, if my Q squared is equal to 0.3, how do I get this to equal just Q? Well, of course, I'm going to square root both sides, right? So I just square root my Q squared. Okay, as I put over here, I take my square root of my Q squared to get Q. So let's do that. All right, so square root of 0.3 is going to equal my Q. Remember, 0.3 is equal to Q squared. So to get Q, you square root both sides. So take my calculator, and I'll do square root, second function, of 0.3, close my parentheses, and I get, okay, Q is equal to 0.5477. Okay, that is what? The frequency of all of the Qs in the population. Not individuals, all the chromosomes that are in living organisms that are carrying the Q or recessive or broken allele. What's the other possibility? Be carrying the what? The dominant, the P. And what do we know? Changing my ink, because you can do it. Okay, what do I know? Well, I know P plus Q has to equal 1. Why? Because that's all the possibilities. So if I know my Q is 0.5477, right? How do I get P? Well, subtract from 1. 0.5477. I should do 1 minus that. Okay. And I get 0.45, say 23. And if I was to, what is this equal to? This is equal to my P. And I may have rounded differently. I think I'm okay. If I add these together, it should give me 1. So all I did was take my Q, which I got from my homozygous recessive individuals that are Q squared. So to find Q, I square root to find the frequency of the recessive alleles. And I subtract it from 1 because x plus this equals 1. So solve for it algebraically. You're going to subtract that. You got the P, because this plus this equals 1. And now I can do this math. I have my P's and my Q's. And now I can go and solve and plug back into the formula. So let's go do 4, 5, 2, 3. And my Q is 0.5477. And why do I have that in green? I want to keep telling you we get this one first. Because this came from what? What did Q come from? If you understand this, boy, you are really, or girl, you really are on to something. It came from my Q squared. What is Q squared? Oh, that's Q and Q. Isn't Q recessive? Oh, that's T and T. That's individuals. So by counting our individuals that are what? Inflicted. People that don't taste the bitterness, we're going to call you inflicted, sorry. Okay, only because I'm a taster. Sorry, but in any case, we can see in society, who is recessive by noticing that they don't have a working gene. So there's some issue, unfortunately, possibly a disease. Now, being a non-taster is not a disease, but hey, I'm a taster. But the point is, I'm the cure. The point is this, T and T. We can see this. In fact, let me just draw an eyeball. So we always start, that's a crazy eyeball, I'm looking, see these guys and count them. We weren't able to count who was homozygous recessive. We weren't able to count who was homozygous, who was a heterozygote. We can't count those genotypes. Okay, it's important that we can't do that. What is the p-squared, people? Well, we kind of know what it is already because we started with that, so that's 0.3. But if you wanted to work backwards and you had, I'm sorry, p-squared we don't have. There's my first mistake. We have the q-squared, people. So we started with this with 0.3. And if you didn't trust me on any of this, and I wouldn't trust myself after that mistake, that this would be 0.30, and I would take my q and square that. But that's going backwards. We started off, okay, with this individual, the q squared. So who's my p squared? What is the frequency of individuals that are homozygous dominant? Don't lose sight what this means. Remember, p is what? Big T. Q is little t. So p squared is big homozygous recessive. These are my heterozygotes. And this, of course, we just found out from our data. Our data gave this homozygous recessive. Okay, so let's go find p squared. All right, so wherever I do with my calculator. So I already have my p. Don't forget how we got p. We always get our q squared first. We did the square root to get this, and we subtracted from 1. And now we're going to do p squared. So I'm just going to take my 0.4523, and I'm going to times that by itself. It would have been easier if I just hit the x squared button, but I didn't. So I get 0.20, don't care about rounding here, 0.2046, we'll just round to 4, we'll just keep it like that. And then my 2 PQ individuals, notice the 2, so I'm going to take, I'm going to find all my heterozygotes.
At this time, we will enact our first lockdown drill. Again, at this time, we will enact our first lockdown drill. The school is now in lockdown.
All right, so we have our p squared, which is a homozygous dominant frequency. That's like 20% of all people. Good, that we got from our jumping off point. Let's get my 2pq. All right, so I know what my p is. So this would be 2 times, oh, Christmas in July. So we're going to go 2 times. My p is 0.4523. And it'd be great if I wrote it that way. All right, I can do this. 0.4523. And I'm timesing it by the what? The q, which is 0.5477. I definitely did not give myself enough room, so all I'm doing is 2 times this times this. Let's put that in there. Put this in my calculator. So 2 times 0.4523 times 0.5477. And if I'm not mistaken, and there's nobody here to correct me because, boy, I'm going solo now, the frequency that I get, okay, let's keep that, whatever color that is, 0.495, let's go 7. Okay, and so if you do a eyeball, shouldn't all the individuals equal 1? Right? Isn't that formula right there? So shouldn't all this equal 1? Absolutely, right? So don't do the individual alleles, do the p squared, so 20 plus 50, 70, 70 plus 30, it's 100. That makes sense. These are all the frequencies of the individuals here. Okay? And that, of course, is the I guess I should have put the calculations above and the numbers below. So there are my frequencies here. All right, so I guess I didn't follow my own worksheet. Okay. Now, interesting enough, how many people are in the class? Well, we know 0.30 because that was our jumping off point. 30% of the class were non-tasters. They were the homozygous recessive allele. But how many people in the class, based on Hardy-Weinberg and based upon no evolution, do we have? Well, take your 0.49, 5.7 frequency, times it by the 20 students we had in the class. Right? Because if you look back on our original work, okay, we had 20 students, right? It was out of 20 that we did our work from, right? So, back to here. And so, you take this frequency and times it, so 0.49 times that by 20. And I get 9.9 students, which is going to round off to 10. Okay, because you can't have 0.9 of a student. So I got 10 students. Okay, this will not be 0.3, 30% of my students, right? So 0.3, I can do this. 0.3 times my 20, right? If you want to do that. That was my 6, right? And then, of course, do the math. Okay, what's 20% of 20? So 0.20. So this one is 0.2046 times the 20 students. That's how I'm getting the total number of students. Out of the total number, remember, this is the frequency times the total number of students gives me the actual number. That's what we're doing here. So 0.2046 times 20. And I get 4.092, which rounds to 4. And lo and behold, 6 plus 10 plus 6, of course, that gets me a total of 20. So what Hardy-Weinberg gives us is, although we know already 6 out of the 20, the 30% were non-tasters, it's actually giving me the percentage of what the other genotypes must be if we're at no, what? No evolution going on. That's pretty cool. There was 14 total, but only 4. Most of them are heterozygotes. Look at that. Is there a heterozygote advantage that still persists in our, what? In our population? Maybe. Now if you go to the last page, okay, go to the last page, and it's really interesting to see what the frequencies of the PTC tasters are. Okay, look at Australian Aborigines. These people are the ones that have the least amount of gene flow. They've been together as a group longer than other people, so they have a higher non-taster part than the Australian Aborigines have a huge non-tasters. Look at the kind of the error bars here. They're not really error bars, they're the mid-range, the percentiles. So this could be, if you think about this, the 25th and 75th percentile. If these are medians, these would be the boxes, right? So these could be box plots. Very interesting. Notice how small that box would be. There's almost no variability. All of them are 49.7. They have so few gene pool, okay, they don't have any genes mixing in, so they're about 50% are non-tasters. What does that mean? Well, we're not really sure, but that's a selective advantage, I guess, okay? Whereas the other people have much lower, okay? And look who has the lowest, Central America, or the Caribbean, okay? And again, you could try to make some sense out of that, all right? And again, if you look at this, early studies show that the ability to taste or related chemical was under genetic control is probably a simple Mendelian trait. People have argued that there's more complexity. Why this trait is maintained in human populations is unknown, but several studies have shown this polymorphism. Okay, remember, polymorphism is basically a change in the DNA called a mutation, which is the non-tasters, right? May influence food selection, nutritional status. Not really sure to make of this, but it's interesting that the Aborigines of Australia, okay, have the greatest percent of non-tasters, okay? And so they don't taste bitter. And maybe that gives them an advantage to eat food that's better for them. But does that give them a disadvantage against the poisons? Maybe, okay? They, of course, live more in a less modern society, and so they have probably a selective pressure, making them have a greater amount of non-tasters. We don't live under that same, or most of these people here live in a modern society, so therefore the selective pressure exists here, okay? And you can stop and think about what that may be, but my guess is being non-taster is there's a selective pressure here to keep them this high, all right? And of course no gene flow there. And what that may be, okay, my guess would be that it allows them to eat food, since they don't get food shipped in like we do, get kiwis and bananas from different places around the world. They eat what's in front of them, and that allows them to eat foods that make them, even if they're bitter, still good for them, and therefore survive better by not having that bitter gene. That's my guess. Hope this helped.